Hey everybody, welcome back to another video. In today's video, we are going to talk about an introduction to the blood. So what are the functions of your blood? What is the components of your blood? And, how do your blood carry oxygen and so much more? We'll dig into a introduction into all of these. If you learned something from the video, if you truly enjoy it, consider leaving a like, maybe subscribing, maybe leaving a comment if you have a question or something. But without further ado, let's get started. So, the blood. What are the functions of your blood? What can your blood do? Well, your blood can carry things. Okay, what can it carry? It can carry gases, such as oxygen. You know that one. It can carry hormones. Okay? Hormones, all of your hormones, in fact, are going to have to circulate in the blood before they get to where they need to go. And fun fact, your blood is actually characterized as a connective tissue. Okay? Your blood can also carry metabolic waste products. Okay? And, there are many examples of this. For example, urea. Another one is creatinine, CO2, many things. And your blood also can carry heat to desired locations, okay? And that's because your blood is around 37 degrees Celsius, okay? So it's pretty hot, and, blood can be redirected to certain areas to maintain temperature, which actually brings me to the next point, which is that your blood is good at regulating temperature, okay? And it also has a function in maintaining proper pH, okay? In fact, the pH of the human blood is somewhere around 7.35 to 7.45. It's maintained within this range very tightly, okay? And we'll take a look at how some of the mechanisms are for maintaining pH, okay? The next one is protection, okay? Your blood can offer protection to you in terms of carrying these cells that can initiate clotting and carrying immune cells and these other things called immunoglobulins. Now, I'm sure most of you know what this is. This is just a fancy term for an antibody. An immunoglobulin and an antibody are the exact same thing, interchangeable. And if you're curious, here's what a blood clotting kind of looks like in a schematic form. So here, for example, is your skin. Here's that layer in between the skin. Here's some fat, and here's the blood vessel. This is what we really care about. All right. Now, if we take a look here, there was some sort of puncture in the skin area. You got your red blood cells flowing through here, right? But you don't want them to kind of escape out, and you don't want anything to come in. So these white cells here, that we'll talk about in a second, called platelets or thrombocytes, they're going to come and plug this hole by forming a clot, okay? So they're going to plug this hole so nothing can escape or come in, and that's called a clot. And, later on we'll see that a clot, on top of it, has like this little kind of stringy, fibrous mesh, and that's actually to hold it in that place, and so this clot doesn't actually get lodged off and move somewhere else, right? Because that'll make things worse. And this thing that is on top of it to hold it there is called a fibrin, okay? It's a protein called fibrin. Anyways, let's move on. So, now that we know the components of the blood, let's actually take a look at what is in your blood and what does your blood look like. So I'm sure that most of you, if you've gone and given blood or, done a blood test, you'll first notice that it's all just kind of red like this, okay? Maybe a little bit of kind of yellow at the bottom. But if we take this guy and we centrifuge it, which means that we just put it in a device that spins it extremely fast, all right, this blood will start to separate into its different components based on their densities. And here's what we would see the blood looking like, okay? It would separate into three portions. One is this big one here, one is this middle layer that's pretty small, and one is this kind of intermediate layer. What are all of these layers, and what are they called? Okay, so this first layer is called the plasma. And you know what? Let me change colors here into this. This side is called the plasma. Now the plasma is pretty much just water, and we'll talk about that, but it's made up of some other stuff as well. But this guy is about 55% of what your blood actually is. It's just plasma, okay? The other part is this green dude here, this green layer, and this is less than 1% of your total blood. And this guy's called the buffy coat. And your buffy coat, we'll talk about it, it contains your white blood cells and those platelets that we talked about that can form those clots, okay? And then here's this last part, which actually contains your red blood cells, okay? So let's bring this bad boy out here. Let's put this here, okay? This guy contains your red blood cells, also known by their fancy name called erythropoiesis. Okay, so cyte means cell, erythro means red cells or red blood cells. Okay, now these guys are 45% of your total blood volume. Okay, alrighty. So now that we know what happens when you centrifuge the blood and you can actually kind of see what the constituents of the blood is. Now, you have to understand that when the blood is kind of running inside of your capillary, it's not like this. It is all kind of mixed together, and the major biggest component is the plasma, okay? Now, speaking of all this stuff, let's kind of go into what is inside of them in order, okay? But first, let's kind of talk about the buffy coat because it's the smallest one, and we'll have videos on this. So the buffy coat contains your white blood cells, otherwise known as your leukocytes. Again, leuko means white, and cyte means cell. Okay. Now I'll tell you something very cool. These guys are not actually white. They don't actually have a color, so they're just called whites. They're pretty much transparent almost. Anyways, the buffy coat, on top of having these white blood cells, I also told you that it contains platelets. Okay, platelets. Now, why is it less than 1%? Okay, well, it's because at a normal homeostatic state, you don't need that many white blood cells kind of circulating inside of your bloodstream. But what if you were sick with the flu or something and you had to go and get a blood work done? Well, in that scenario, this buffy coat, if we were to measure, it would actually be larger than normal, okay? It'll be greater than 1%, right? Because you're sick and you have those white blood cells in your blood circulating more to try and defend your body, okay? Pretty interesting. Alrighty, so that's the little tidbit about the buffy coat, right? Now, the combination of the buffy coat and the erythrocytes, right? So if I put that 45% and that less than 1% together, is called the hematocrit, okay? And it's about 45%, you know, like it changes. It's about 42% in women, 45% in men, okay? But this is the hematocrit, and all it's talking about is just formed elements, okay? And like I said, it's the combination of the buffy coat and the RBCs. Alrighty, now let's talk about the plasma. This is the biggest component, so when we get this out of the way, the rest of the video is going to be smooth sailing, I'm telling you guys. Now, like I told you, the plasma is pretty much 90% water. 90% water, okay? So this just goes to show you that your blood is pretty much a lot of water. The other bits include these things. So there's about 3% of what we classify as other. But what the hell is other? Well, other is these things. Other includes nutrients, waste products, right? Remember those waste products we talked about over here? Okay. Gases, right? Remember the oxygen, the CO2, all of that, and hormones, okay? So all of those stuff that we said are going to get carried, right? They're all inside of the plasma. They're dissolved or being carried by something, some protein within the plasma. Okay. The other portion of this other is electrolytes. Okay. Electrolytes. And your most abundant electrolytes inside of your plasma are actually going to be your sodium and your chloride ion. Okay? So remember, chloride has a negative charge, sodium has a positive charge. Okay, then pretty abundant inside of your plasma. All right, so that's 93%. What about the other 7%? Well, the other 7% is just going to be, let's do it like this, 7% plasma proteins. And it's really as simple as it sounds. These are just proteins that are freely floating inside of your plasma, and we can broadly categorize them into three groups. Okay, three groups. One of them is going to be this dude called fibrinogen. Now, this term fibrin should sound familiar to you because I mentioned it. And fibrinogen is actually the inactive precursor for a blood clot's fibrin meshwork. Okay? All right, so remember how I told you that when a blood clot is formed, in order to keep it stable and keep it there, this fibrin kind of stringy material has to come sit on it? Well, that fibrin is originating from this guy, fibrinogen. Okay? Fibrinogen is just an inactive precursor of fibrin, right? Great. But, during situations where a blood clot is needed, this fibrin will become activated and turned into, fibrinogen will be turned into fibrin. Okay? Great. And in fact, other times when you go and get your blood, or you might be seeing it somewhere, you might hear the term serum. Okay, serum. Okay, or actually plasma without fibrinogen inside of it is called your serum. Okay, so it's literally just your plasma serum, but just without fibrinogen. All right. So no kind of clotting, stabilization, or anything like that. Okay, great. Let's look at the other portions. Okay, so this is one plasma protein, fibrinogen. It's just a protein. The other thing is albumin. This big guy over here, albumin. Now, let me pause over here. Let's pause over here. Remember how I've mentioned this in other videos. Proteins that are inside of the plasma in general, they will classify as non-penetrating solutes, meaning that they can't cross the red blood, the capillary or blood vessel membrane. They're just too large, okay? So they would be like these proteins that are just kind of in here, and they're too large to kind of pass through the blood vessel. These guys, because of that, they all proteins, they all contribute to the osmotic pressure of the plasma. All proteins, okay? Some, however, contribute more. Albumin, for example. Albumin is the biggest contributor to the colloid osmotic pressure. By the way, colloid osmotic pressure is just referring to the osmotic pressure originating inside of the plasma, okay? Because, they say that the plasma proteins inside of the plasma form this thing called a colloid. You don't really have to worry too much about what a colloid actually is, okay? Just know that they form a colloid. It's like a gelatinous solution, okay? Like the whites of the egg, right? There you go. You can think of it like that. But albumin by far is the biggest contributor to the colloid osmotic pressure, while fibrinogen and all the other plasma proteins do play some role as well, okay? But albumin is the biggest contributor, and there's a reason for this. It's because albumin is the most abundant protein inside of the plasma by far. Like, this guy is there's so much of this guy, okay? Now, what does he actually do? Well, aside contributing to the colloid osmotic pressure, he's also involved in the transport of transport of non-specific substances. Okay, what does this mean? This means that this guy is able to transport things that are not soluble in water, because the plasma is 90% water. So this albumin molecule, this protein, he can transport things that are not water soluble. What kind of things? Things like free fatty acids. Okay, so fatty acids can be transported easily by this guy. In fact, albumin has 7 binding sites for fatty acids. It can transfer other things like micronutrients. Certain hormones might bind to albumin. There are some forms of testosterone that bind to albumin. But again, it's very non-specific in what it transports. It doesn't really care. It's just there to kind of help out and transport whatever, right? The other classification of plasma proteins is the globulins. Okay, and the globulins themselves can be characterized into 2 portions. Okay, one is the gamma globulin, and the other one is the alpha and beta globulins. I apologize, my Greek letters are not very nice. But the alpha and beta globulins, as opposed to albumin, they transport very specific substances. Okay, so but again, similar to albumin, they transport water insoluble stuff. Okay, whatever does not dissolve in water insoluble stuff. Okay, but they are specific in what they transport. Okay, and I'll give you a couple examples of these. So I'm sure you guys have heard of like HDL, LDL. Okay, these guys, they will classify as alpha and beta globulins. I don't know how much you guys know, but for example, this iron transfer protein called transferrin, okay, he is also a protein that is inside of the blood, but his entire job is to just transfer iron around, okay? There's other things like, for example, complement factors, okay? But you get the gist of it, okay? They transport specific stuff such as cholesterol, iron, complement via these alpha, beta, and globulins. The gamma globulins, they are literally just another version of calling antibodies, okay? So far we've called them 3 things. We've called them antibodies, gamma globulins, and immunoglobulins, all right? Same thing. All right, look at this. The plasma does so many damn things, guys. So many damn things, all right? And remember how I was talking about that pH thing before? Well, guess what? These proteins, okay, not only do all of them contribute to the osmotic pressure, the colloid osmotic pressure, meaning the plasma's osmotic pressure, okay, which, by the way, is extremely important in the distribution of fluid between the blood vessels and the interstitial space which surrounds the cells, okay? But these proteins, they also play a role in buffering any pH changes. So if the pH, for example, becomes very like acidic, okay, these proteins can lower that pH and make it more basic again and bring it back to that range that we were talking about of 7.35 to 7.45, okay? So they're pretty important in these kind of functions. Now, of course, they're not the only thing that contributes to that pH thing, but they're one of the things that do, okay? Alrighty, so now we talked about plasma, we talked about the Buffy coat. Now let's get into this guy. He is what you want to know, the red blood cell. Okay, the red blood cell, guys. This red blood cell, man. It has so many cool things in it, okay? So here's, first of all, an image of the red blood cell. Right here. Here's this red blood cell, okay? And I have a picture, I have the symbols of men and women here, okay? And it's just really supposed to represent the blood volume, okay? So for men, we have a blood volume of about 5.5 liters, whereas women have a blood volume of 5 liters, okay? Again, these are averages. And this has nothing to do with the red blood cell, it's just talking about the blood volume. But let's get back to the red blood cell. The red blood cell right here, this is its shape, okay? There are approximately 250, there's about 5 billion red blood cells per 1 milliliter of your blood, okay? And so you can imagine that if we have 5 liters of blood, or 5.5 liters, then we have trillions, right? Like 25 to even 30 trillion red blood cells inside of our body. That's an insanely huge number, okay? But what do red blood cells really do? Well, they're best known for carrying oxygen, okay? And we'll talk about that. But first, let's give a little few facts about these red blood cells. First of all, is that these red blood cells, they live only about 90 to 120 days, okay? And there is a reason for this, which we'll talk about, okay? But for now, just remember that they live about a 90 to 120 days, and when they die, okay, the old and damaged red blood cells are removed by an organ in your body called the spleen, okay? In fact, the spleen has these special cells called macrophages that will go ahead and remove these guys, okay? Now, the structure of the red blood cell, again, is this kind of circular shape, but you'll notice that in this middle, it has like this kind of donut shape, okay? They call this like little area where it's gone inwards. They say that this right here is a biconcave disc, okay? And if I were to draw a red blood cell like this, look at this, okay? You'll notice it's something like this, where the inside portion is kind of like flattened out, all right? Perhaps you can see it better over here, okay? There you go. You can see it right here. This portion over here seems to be flat. Why is that the case? So why would we want to have this biconcave disc? What does this do for us? Well, this biconcave disc, turns out, increases the surface area of the membrane for gas exchange. Because you know, one of the main functions of red blood cells is to exchange oxygen and CO2 at the lungs, right? And at the level of the tissues. Well, if I increase the surface area of this actual red blood cell by creating this biconcave disc, then I'm increasing the amount of gas exchange that I can do, okay? That's just fixed laws of diffusion 101. Go and watch my membrane video if you haven't. Okay, so that's why we want this biconcave disc. Now, what's the big idea though? How can they carry oxygen and all these other gases? Well, red blood cells, they are like an extremely specialized cell. They're pretty much just bags of this thing we call hemoglobin. Okay? This is what hemoglobin looks like, guys. Right here. This molecule, this polypeptide, which is just a fancy word for protein. This guy's hemoglobin. So watch. Hemoglobin. Now, hemoglobin is the molecule that's able to transport oxygen, as among other things as well, which we'll talk about. Here's what it kind of looks like. It's made up of 2 alpha globin chains, and these are just proteins. Globin peptide chains. Remember, peptide just means protein. And another subunit called beta globin peptide chain. Now, if you're curious, technically this would be beta 1, alpha 1, and then this would be beta 2, and then this one would be alpha 2. Okay? So in total, it is made up of 4 globin peptide chains. 2 alphas, 2 betas. Now, each one of these guys, okay, contains this molecule inside of it. This molecule right here in yellow, which is called a heme group. Now, heme is a pigment, and if you're curious, it's actually a red pigment. This is why our red blood cells appear red, and this is why our blood appears red. It's because of the presence of this molecule called heme inside of hemoglobin. So you can see that it's a pretty, rightfully named structure, but the globin chains and the heme molecules that each globin chain consists of. So one hemoglobin molecule is consisted of 4 peptide chains, each one consisting of 1 heme group, so 4 in total. And in the center of this heme group, there is this iron, okay? This iron that really, when we say hemoglobin binds oxygen, it is really binding to this iron, okay? So each one of this guy could, for example, carry oxygen. O2, 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 and O2. So if I were to ask you how many oxygens does one hemoglobin carry, you would say 4 oxygens. Okay, and that's because of the presence of these iron-containing heme groups. Beautiful, beautiful. So that's hemoglobin, right? That's hemoglobin. Now, there is about, and this is going to be crazy to know, there's about 250 million hemoglobin molecules per red blood cell. Okay, per RBC. All right. That means that, and you know, 250 million, and each one can carry 4 oxygens. That means that 1 billion molecules of O2 are being carried per red blood cell. Like each red blood cell is carrying 1 billion oxygen molecules. That's insane. That's so much. Okay? Now, let's learn some other things about the red blood cell, okay? So we know that it is just a bag full of hemoglobin, okay? But what else does it have? You know this red blood cell, it has a lot of glycolytic enzymes. Glycolytic enzymes. Now, what are glycolytic enzymes? Let's think back to my glycolysis or metabolism video. I said that glycolysis has to occur inside of the cytosol, and that means that it does not require anything about mitochondria or any other kind of thing. It just occurs in the cytosol, and there are enzymes that help assist the process, right? Now, a fun fact about the red blood cell is that, remember I told you it lives only like 90 to 120 days? Well, that's because it can't actually go through cell division. You might be asking, hey Keon, why doesn't it go through cell division? It can't go through cell division because it doesn't actually have a nucleus. That's right. Or any organelles for that matter. Well, most organelles. During the maturation process of this red blood cell, it actually ejects its nucleus, quite literally ejects its nucleus outside, and it gets rid of all of its organelles, such as the mitochondria, so it can't go through oxidative phosphorylation to produce ATP or energy, which obviously every cell needs. So it can only rely on the glycolytic enzymes found in its cytosol. That's why it has a high concentration of these guys, okay? And that's why it can only live not 90 to 120 days, because it can't go through cell division, because it has no nucleus. Before we talk about this other thing, let's say what else hemoglobin can carry. Hemoglobin can carry this molecule called nitric oxide, which is able to vasodilate your blood vessels. Okay, so nitric oxide is another gas that can kind of bind to this. We know that it carries oxygen, and in fact, oxygen is extremely insoluble in water, and 98.5% of oxygen is carried in the form of hemoglobin, and the other 1.5% is kind of dissolved inside of the plasma. Okay? All right. CO2 is also carried by hemoglobin to a lesser amount. CO2, as we'll learn in a second, is carried in different ways, multiple different ways. And if you're curious, I have a whole video on oxygen and on CO2 transport in my Human Physiology Part 2 playlist, so feel free to go and watch that. It can also bind to carbon monoxide somewhat irreversibly. Okay, so in places, certain textbooks, you might see it be called hemoglobin binds irreversibly to carbon monoxide. That's not entirely true. It binds with a much more higher affinity than oxygen, but you can still kind of get it off or force it off of hemoglobin, right? But you don't want carbon monoxide, obviously, over here, because it offloads oxygen, and that means you're not going to get any oxygen to your body. The last thing is protons, or these hydrogen ions. They can also bind to hemoglobin, okay? And I'll tell you this, the presence of protons inside of the bloodstream dictates the acidity. So if there's a high concentration of these protons, the blood is very acidic. Now, remember that I said that many things inside of this bloodstream can kind of tie up or lower the acidity, one of them being proteins, okay, those plasma proteins. The other one being this hemoglobin. He is able to actually bind to some of these hydrogen ions, or these protons, and prevent the blood from becoming too acidic, Okay? Great. So those are the things that hemoglobin can bind to. The final thing that you will find when you look inside of this red blood cell, right? Remember, we said that you can find hemoglobin, you can find glycolytic enzymes. The final thing is this other enzyme called carbonic anhydrase. This guy is so important. He catalyzes a very, very important reaction. And this important reaction looks just like this. We're going to take CO2 plus water, okay? And we're going to create H2CO3, which is carbonic acid. And then the H2CO3 being an acid will dissociate into bicarbonate plus a hydrogen ion, okay? Remember how I said CO2 is transported a little bit differently than oxygen? So some of the CO2 can actually be bound to hemoglobin. The other parts of the CO2 actually go through this reaction. So where does CO2 get produced? It gets produced at the tissues, right? It's a metabolic waste product, and we have to get it into the red blood cell, or the blood actually, and then transfer it all the way to the lungs so that we can breathe it out. Well, once CO2 enters the blood and goes into this red blood cell, right? It goes through this reaction and it goes this way. The CO2 inside of the red blood cell will combine with water, and in the presence of this enzyme that we called carbonic anhydrase, this guy right here, this reaction will continue, where CO2 and water will form carbonic acid, H2CO3, and the carbonic acid, being a good acid, dissociates into this base called bicarbonate and protons. Okay, now this proton can go and bind to that hemoglobin over there. All right, but this bicarbonate, he is carrying our CO2. Okay, he's basically kind of like a placeholder, right? Until we get to the lungs. When we get to the lungs, this reaction can actually be reversed. Okay, it can go this way now. At the lungs, we will combine a proton plus this bicarbonate, and we'll turn it into carbonic acid, right? And then this enzyme over here will catalyze the conversion of carbonic acid into water and CO2. And then the CO2 can be dropped off at the lungs and then exhaled. Now, that's the function of this kind of carbonic anhydrase, and all of this entire reaction takes place inside of this red blood cell. Okay? Alrighty, guys. So now you know a lot about the red blood cell and about the blood and its components. Okay? Now we have to talk about, if these red blood cells truly live only 90 to 120 days, then how is it that I have 5 billion RBCs per 1 mil of my blood? Or how is it that I have 25 to 30 trillion of them per my total blood volume? Well, that's a great question. And since we know that these guys don't have a nucleus and they can't go through cell division or mitosis, they have to be made somewhere. And they are. Inside of the bone marrow. Okay? Inside of the bone marrow, specifically the red bone marrow. In your long bones, okay? These guys go through the process of red blood cell formation, okay? Red blood cell formation, or erythropoiesis, right? Now, what ends up happening is there is this specialized stem cell inside of the bone marrow called a pluripotent stem cell. Sometimes you might see it called a hematopoietic pluripotent stem cell. Either way, this guy will start differentiating to become a red blood cell through certain conditions, which we'll talk about in the next slide. But essentially, this guy can become a myeloid stem cell. Okay? This myeloid stem cell can then become something called an erythroblast. Now, notice what's happening here, okay? Notice what's happening here. At this stage, we have a nucleus, have a nucleus, and we have a nucleus here. Past this point, okay, we form this molecule, this more mature version of this erythroblast, which is called a reticulocyte. Notice that this guy does not have a nucleus, so that means somewhere from here to here, this guy must have ejected his nucleus, okay, and it must have destroyed all of its remaining organelles, right? The reticulocyte is the stage right before a mature red blood cell or an erythrocyte, okay? In fact, as soon as this guy gets released into the bloodstream, he will now be called an erythrocyte, okay? So it all started out in the red bone marrow with this hematopoietic pluripotent stem cell. Pluripotent just means that the stem cell can turn into a lot of things. It has a lot of potential, okay? Now, here's the thing, though. Here's the thing. It doesn't just give rise to the red blood cell, guys. This hematopoietic stem cell, it gives rise to all of your blood cells. All of them, including your white blood cells and your platelets, okay? And in our next video, when we start talking about, the immune system, we'll start talking about how this same dude will turn into a myeloid stem cell and give us many different white blood cells. Okay, so this is kind of the differentiation pathway for the red blood cell, right? But there are different differentiation pathways for different blood cells, all right? That's important to remember. Now, if you're really curious, we actually did skip a couple of important steps just for the sake of simplifying the video, okay? But for those who do want to know, after this myeloid stem cell, we actually get something called a pro-erythroblast. Then we get something called a basophilic erythroblast. Then we get something called a polychromatic erythroblast. And then finally we get something called an orthochromatic erythroblast. And that orthochromatic erythroblast is the guy that's going to eject its nucleus, and then we'll turn into that reticulocyte, okay? Again, you don't have to know this. This is just kind of a little bit of extra information for the people who want it, okay? So that was the process of erythropoiesis, or red blood cell formation. But now let's see what can stimulate this process, because it doesn't just happen on its own, okay? And, and there's about a 2 to 3 million red blood cell per second turnover rate, okay? So it's happening pretty frequently, okay? But what is the stimulus? So let's take a look at what the stimulus is, okay? This is actually very, very important. Now, first things first, let's change the color here, and boom, there we go. So we start out at the kidney, okay? Notice how there's only 2 red blood cells here. The kidney is going to sense a low oxygen carrying capacity. What does this mean? What does a low oxygen carrying capacity mean? Well, if you have a low number of red blood cells, right, that means that you have a low number of hemoglobin in total, okay, lower than your normal amount. That means you're carrying less oxygen than normal. That means you have a low oxygen carrying capacity. Now, there are special cells inside of this actual kidney they're called renal interstitial fibroblasts. You don't have to know that, okay? Just know that there are specialized cells in the kidney that can sense whenever there's a low concentration of oxygen being carried in the bloodstream. And when they sense this, okay, the kidney starts to secrete a specific molecule, a hormone, if you will. This hormone is called EPO, which stands for erythropoietin, okay? And erythropoietin is these kind of green molecules. Now notice, there's a lot of erythropoietin in the blood right now, but there are still those 2 red blood cells that came through this kidney and then passed through that kidney, okay? So these guys will continue until they reach the red blood cell, red bone marrow. This erythropoietin, this hormone, it'll stimulate the process of erythropoiesis. This process right here, it'll stimulate that process inside of the red bone marrow, and after the red bone marrow is done, it'll release those mature reticulocytes into the bloodstream, which will now be called red blood cells or erythrocytes. Okay? So, one more time, the EPO stimulates the red bone marrow to make red blood cells go through erythropoiesis. And you would agree with me that erythropoietin then is rightly named. And these guys, look at them. There's 1, 2, 3, 4, 5 red blood cells as opposed to the initial 2. Now your oxygen carrying capacity is pretty high. And so these guys will continue, and they make their way to the heart, obviously, and then they get pumped out. And the next time they reach the kidney, guess what? Those kidney cells that sense low oxygen carrying capacity, they're going to be like, oh, okay. The oxygen carrying capacity is back to normal because now we have a lot of those red blood cells, right? And thus, the initial stimulus is going to be inhibited. And these guys will no longer produce erythropoietin, and this cycle stops. This is a classic example of something called a negative feedback loop to maintain homeostasis inside of the body, okay? Let's review the steps. Step 1, kidney senses low O2 carrying capacity, okay? 2, kidney increases EPO in the blood. 3, EPO stimulates the bone marrow, okay, to go through erythropoiesis. And then 4, the O2 carrying capacity is back to normal. Back to normal. Which does what? Which actually inhibits release of EPO. Okay, and that's called a negative feedback loop. So that's one situation where erythropoiesis can happen, okay? And you might be wondering, hey, when do we have low O2? Under situations like hypoxia, okay? Hypoxia is low oxygen in the blood. For example, when you go up onto high altitude, right? This can definitely occur. Okay, guys, that's going to finish it for this video on the introduction to the blood. Next video, we're going to talk or have a brief discussion about blood typing, and then we'll move on to the white blood cells or our immune system. If you enjoyed the video, if you liked it, if you learned something from it, please leave a comment, like the video, and subscribe, and I'll see you next time.